Okay, continuing with heart. We're looking at the microscopic anatomy of cardiac muscle. So this is the histology of cardiac muscle. It was covered last semester in tissues, briefly. It has similarities to skeletal muscle tissue, but there are some significant differences. Because the cardiac muscle has to contract once a second for 100 years, it is a huge consumer of energy. It needs a lot of ATP, and that ATP has to be done aerobically because you get 36 ATPs then per glucose. That means a lot of oxygen has to be supplied and relatively a fair amount of glucose as well. And there has to be a lot of mitochondria because, as you remember, Krebs cycle happens in the mitochondria. There is myoglobin, which is the pigment that stores oxygen in red meat, in red muscle. It's the same as hemoglobin, except it's found in muscle. It has iron in it, and it stores oxygen. The heart has a huge amount of myoglobin. That's why hearts are really deep red. It is very efficient at producing ATP because of this energy. It can oxidize fats, glucose, lactic acid, amino acids, ketones. Just about any organic molecule can be oxidized by the heart and turned into ATP to continue beating. The heart's very robust against any of the waste products of those things. For instance, when you use amino acids, you have nitrogen left over. That's in sharp contrast to neural tissue, which is also metabolically very active, but can only burn glucose because it can't tolerate the nitrogenous waste and the lactic acid waste, etc. So the cardiac muscle is a striated muscle with myofibrils in it. A bands and I bands, and like all the little sarcomeres are the same. There's troponin and tropomyosin and actin and myosin, and the same cross-bridging happens. It requires calcium, the same sort of thing. I've posted some animations of this stuff, and it's worth watching. The cells themselves, like I say, are striated, but they are branched. They are also they're non-voluntary. Like they're going to contract whether you think about it or not. So they're similar, but there's way more mitochondria. They're branched. They join onto each other in this space called the intercalated discs. And the intercalated discs have desmosomes to hold the cells together and gap junctions so that the cytoplasm of the one cell can mix with the cytoplasm of the other. We'll talk about that a little bit later. There's a huge need for oxygen, which means there needs to be a blood supply. Now, the heart doesn't take any oxygen from the blood that it's actually pumping. Instead, there are arteries that are the very first branches off the aorta, branches for systemic circulation, that bring oxygenated blood via arteries to the myocardium of the heart. And we call these the coronary arteries. We generally, when we refer to the heart's function, we say cardiac. When we refer to the heart's anatomy, we say coronary. So the coronary arteries transport oxygenated blood from the aorta to the walls of the heart. And then the coronary veins come from the capillaries that are supplied by the coronary arteries, and they collect the deoxygenated blood and they bring it back towards the right atrium via the coronary sinus. So when we look at the coronary arteries, there's a fair amount of variability, but most people have a left and right coronary artery coming off the aorta, right where the aorta emerges from the heart. The left one over here, the right one over here. Now, this groove between the atrium and the ventricle is called the coronary sulcus. It goes all the way around the heart there. And then there's a groove that's in the front of the heart over top of the interventricular septum called the interventricular sulcus. So the left coronary artery emerges and then branches into the anterior interventricular artery, which goes around to the apex in the anterior interventricular sulcus, and the circumflex artery that bends around behind the heart in the coronary sulcus. Circum, going around, flex, bends. So this is the artery that bends around to the back. On the right side, we have the right coronary artery that comes around here. There's various branches, and these all have names, but there's one major branch called the right marginal artery that goes down the right margin of the entire heart. Now, all the little branches off of these ones feed the walls, the myocardium, front and back of the ventricles. The right continues around here and then ends up going down in the posterior interventricular sulcus, so it's called the posterior interventricular artery. Now, usually the posterior and the anterior interventricular arteries meet up so that they've got a detour, a backdoor, that's called an anastomosis. There's usually an anastomosis here, and there's usually an anastomosis here between the circumflex and the posterior interventricular. And it gives a detour route for the blood to go if there's a blockage. When they do coronary bypass surgery, it's because there's a blockage, say here. And then they would take a bit of the great saphenous vein and bridge over the blockage. If they have to do one, it's bypass surgery. If they have to do two, it would be double bypass. If they do three, a triple bypass, etc. These backdoors, these anastomoses, are not all that efficient. If there's not sufficient flow to the heart of the wall of the myocardium, then what happens is you get ischemia in that heart wall. Not enough oxygen being delivered to produce the ATP that's necessary. When that happens, the heart starts to produce ATP anaerobically, which produces lactic acid, which then hurts, like any muscle filled with lactic acid. And that's what angina is. And if it gets to the point where there's not enough ATP to actually run the sodium-potassium pumps and to have the heart contract, then that's when you get a myocardial infarction or heart attack. Okay, so the blood flows back through the veins. The one that runs parallel to the anterior interventricular is called the great cardiac vein. The middle cardiac vein is the posterior interventricular. The small cardiac vein runs kind of along with the marginal. And there's various other veins as well, but they all end up back in this thing called the coronary sinus at the back. A sinus is just like a vein, except it has no valves. It tends to be dilated as well. So the blood pools in here and then enters the atrium right here at the sinoatrial opening. Now, there's variability. Not every individual has this. Not every individual has the same pattern with the coronary arteries. There's more variability with the veins than there is with the arteries. Okay, because of the gap junctions, the heart acts as something called a functional syncytium. Functionally, it acts as if it's one cell. An action potential in one cell passes the positive ions, the calcium and the sodium, pass through gap junctions into the next cell, and they depolarize that and cause an action potential when it reaches threshold of the next cell, and so on, till the whole heart contracts on its own as one thing. To be an efficient pump, you want all those cells to contract simultaneously. So this is all coordinated through specialized cells. They're muscle cells that don't contract. They just carry action potentials. They are, functionally, they are the same as nerves. Histologically, they are cardiac muscle cells. And then the autonomic nervous system modifies that. But the heart has this autoregulatory thing. It actually will beat on its own. The heart doesn't need to be hooked up to the nervous system to beat. You've probably seen videos of hearts removed and sitting on a tray still beating. The heart will beat until it runs out of oxygen and runs out of glucose. It's the reason we can transplant hearts. We can't transplant nerves, and so we can't hook up a transplanted heart to the nervous system. The heart beats on its own. And that's the intrinsic conduction system does that. Now, within the heart, well, let's back up. When we talked about muscles last semester, and when we talked about nerves last semester, we talked about sodium channels. Some sodium channels open because of chemicals, ligand-gated channels. Some channels open up because of voltage changes, so that would be voltage-gated channels. And we talked about how acetylcholine will cross the synaptic cleft, bind to the ligand-gated sodium channels, open the sodium channels. Sodium will rush in, depolarize the cell. That depolarization causes the voltage-gated channels to open in succession, which then causes the wave of the action potential, which then goes down the T-tubule, causes the sarcoplasmic reticulum, calcium gates, voltage-gated calcium channels to open. The calcium leaves the sarcoplasmic reticulum, binds onto troponin. The troponin pulls the tropomyosin out of the way, exposing the binding site. Cross-bridge happens, contraction. Well, the same thing happens in heart, except rather than a ligand-gated channel initiating this, there are slow-leaking sodium channels. They are always open. You pump the sodium out of the cell, and it leaks back in. When enough sodium is leaked back in to reach the threshold of depolarization, then the voltage-gated channels open, the positive ions come in, action potential. Then these positive ions pass through the gap junctions into the next cell, and they start a chain reaction. We call the cells that do this, and it's about 1% of the cells in the heart can do this. We call them autorhythmic cells. Now, there's a group of these autorhythmic cells that are all bundled together. They're all sitting right beside one another, and it's a node, and it's right where the sinus enters the right atrium. So we call it the sinoatrial node. And it will actually reach this threshold about 75 times a minute. So sodium's pumped out, it leaks back in, causes an action potential. Is pumped back out, leaks back in, reach threshold, action potential. 75 times a minute. Because of that, this is called the pacemaker cells. Now, as the pacemaker cells reach an action potential, it causes the contractile cells all around it that are connected via gap junctions to start to contract. So a wave of contraction of action potentials will spread across the atria, the two atria, and the atria will start to contract and push blood into the ventricles, the last little bit of blood into the ventricles. There are a couple of these conducting cells that actually go to another node called the AV node. Now, the AV node is right on the floor of the right atrium, right where it's about to go into the ventricle. Now, this node has pacemaker cells in it as well, but they're about 50 times a minute. So they're not normally the dominant one unless something happens to the SA node. What happens at the AV node, it's like a bottleneck, and the action potentials kind of get delayed there by about 0.1 seconds. And then the action potentials continue from the AV node down into the interventricular septum through a group of cells that are called the AV bundle, or sometimes called the bundle of His. When I was a student, I tried to remember that as the histologically interventricular septum. This immediately divides into left and right bundles, and the bundle branches. And so in pathology, we're going to talk about people that have problems with these and called the bundle branch blocks. It reaches the apex and then spreads out along the outside of the myocardium through fibers called Purkinje fibers. And it was Dr. Purkinje that figured this all out, so that's why he gets his name attached to it. And as all of these cells reach their action potentials and have their action potentials, they spread through gap junctions, the action potentials, into the contractile cells, and the ventricles will contract. Now, the autonomic nervous system can modify this. In the brain, in the medulla oblongata, there are acceleratory and inhibitory centers. The sympathetic nervous system will increase the activity at the SA node and at the AV node and make them reach threshold sooner. They'll open more channels, and it will reach sooner, so therefore it speeds up the heart rate. The sympathetics inhibit them reaching threshold, and so decreases the heart rate. We'll see also that the sympathetics increase the force of contraction, but that's neither here nor there. So the sympathetic and the parasympathetic only modify the rate and the force of the heart contraction. It doesn't cause it. The parasympathetic is the vagus nerve, cranial nerve 10, and we often say that the heart is under vagal tone. Most people's heart runs under the parasympathetic and is lower than the 75 beats a minute. Sympathetics increase it and increase how hard it beats. Here's a diagram of it. I'm not terribly worried about this. So the action potential starts at the SA node, comes across through the internodal pathways to get to the AV node. The two atria contract, and when we call the contraction of any part of the heart systole, so this would be atrial systole. We call the relaxation of cardiac muscle diastole. So we end up with a little bit of atrial systole. There's a bit of a bottleneck, and there's a 0.1 second delay at the AV node. So when the ventricles are still in diastole when the atria go into systole. So the last little bit, the last 20% of blood that's still in the atria gets pushed through the AV valves, the mitral valve and the tricuspid valve, into their respective ventricles. Then the action potential goes through the AV bundle and the bundle branches all the way to the Purkinje fibers, and then causes contraction, systole, of the ventricles. The pressure increases and the blood is pushed out of the ventricles through the semilunar valves and into the great vessels. There's a very good animation that I've posted that shows this. So, SA node, depolarization, AV node, bundle branches, Purkinje fibers. We'll do this in the next video.